All right, let's jump right into this explainer. Today, we're unlocking biological systems, and we're going to take this nice and slow, really breaking it down so we can fully digest how tiny living organisms are used to create valuable products on a massive, massive scale. Here is our roadmap for this micro to macro journey. We'll be covering what biological systems are, meet the microbes, how they multiply, surviving extreme conditions, and finally, fluids in the bioreactor.
Okay, Section 1. What exactly are biological systems? The crucial point here is that a biological system is essentially just us using living things to do microscopic manufacturing. Think of it like a tiny living factory. You feed these organisms raw materials, which in the industry we call substrates, and then, through totally natural bioprocesses like fermentation, they transform that food into incredibly valuable products. It's literally living biology put to industrial work. Section 2. Let's meet the microbes, bacteria, yeast, and fungi.
First up, bacteria. These are incredibly efficient, single-celled organisms. We call them prokaryotic. Now, that's just a fancy scientific way of saying they have a cell wall on the outside, but they completely lack a distinct enclosed nucleus on the inside to hold their DNA. Think of it like an open floor plan apartment where everything just happens in one shared space. This super simple structure allows them to have incredibly diverse metabolisms and thrive pretty much anywhere. Next, we have yeast. You actually interact with yeast all the time, like when you're baking a loaf of bread or brewing beer. Now, unlike bacteria, these are a type of unicellular fungi, and they are eukaryotic. This means their interior is a lot more complex. They actually have a true membrane-bound nucleus holding their genetic material, plus other dedicated structures like vacuoles and mitochondria. They are the absolute superstars of fermentation.
Finally, we have filamentous fungi. Unlike the single-celled bacteria or yeast we just talked about, these are multicellular organisms. They have these amazing branching structures called hyphae, and they grow by forming extensive, tangled networks called mycelium. You can kind of think of these networks like a microscopic root system. It allows them to absorb nutrients and produce really critical enzymes for bioprocessing.
So, to make sure this really sticks, let's just do a quick side-by-side. Bacteria are prokaryotes, are fast, adaptable workers that grow incredibly rapidly in a ton of different environments. Yeast, on the other hand, are eukaryotes, unicellular fungi that are highly specialized and absolutely vital for specific processes, like creating alcohol or giving us those fluffy baked goods.
Section 3. How do these microbes actually multiply? The secret to their massive, exploding population growth is asexual reproduction. In this process, a single organism just makes an exact copy of itself. There is absolutely no mixing of genes. It simply creates an identical clone. This allows for breathtakingly fast population growth without any genetic variation. And honestly, that is exactly what you want when you need billions of identical microscopic workers to do a specific job. For bacteria, this specific cloning process is called binary fission. We can break it down into 4 really simple, highly mechanical steps. First, you start with the parent cell. Second, it replicates its DNA. Third, that duplicated DNA segregates, moving to opposite ends. And fourth, the whole cell simply splits into 2. So the parent cell elongates, duplicates that core ring of DNA, a crosswall or septum forms right down the middle, and it just pinches apart into 2 perfectly identical daughter cells. It's a beautifully simple, totally symmetrical split. Now, yeast do things a little differently through a process called budding. It's kind of similar to binary fission, but crucially, it's not a symmetrical split down the middle. Step 1, a small bud appears on the outside of the cell while the DNA doubles. Step 2, the newly replicated nucleus moves into that tiny bud. Step 3, the bud separates and it leaves behind a really distinctive little scar on the mother cell.
You can see how asymmetrical this is. Instead of splitting evenly in half, the mother yeast cell grows this tiny bubble, or bud, on its side. This bud gradually gets bigger and bigger until it eventually just pops off. What's really fascinating here is that the mother cell keeps on going. Both the new offspring and the mother are completely capable of budding again and again, multiplying the whole population incredibly fast.
Section 4. Surviving extreme conditions.
Look, life isn't always easy for these clones. When certain bacteria get severely stressed out by their environment, maybe it gets way too hot or they completely run out of food, they can form what is called an endospore.
They wrap their vital DNA core in these incredibly dense protective layers. It's essentially a way for the bacteria to go into a highly resistant, deep hibernation until conditions become safe again.
Molds and filamentous fungi use spores too. If you think about it, these spores act like microscopic armored vehicles for the organism. They provide extreme resistance against heat, freezing, drying out, and even harsh chemical agents. It guarantees that the biological system survives to germinate and grow all over again when the environment finally gets better.
Okay, Section 5, Fluids in the Bioreactor. Let's zoom out to the macro scale.
So here's a really practical question to think about. How do you actually mix and stir a giant industrial tank full of billions of living, multiplying microbes? It sounds simple enough, right? Just stir it like a giant cup of coffee. But pumping and mixing these enormous biological populations creates incredibly complex physical challenges that engineers have to carefully solve.
This massive, highly controlled tank is known as a bioreactor. It's the industrial home where our microbes actually live and work. And the specific design of this tank isn't just random. It depends entirely on the unique traits of the organism inside it. Things like how fast they grow, what temperature they need, and the physical and chemical nature of the product they are synthesizing for us.
But here is where the physics gets kind of wild. The major problem is that a thick, dense broth of billions of microbes doesn't just flow easily like water. It acts as a non-Newtonian fluid. All that means is that its viscosity, or its thickness, actually changes depending on the shear rate applied to it. In really simple terms, how thick or thin the liquid is changes entirely based on how hard you stir or pump it.
To make this perfectly clear, let's look at some everyday examples. Some microbial broths are pseudoplastic. They actually thin out and get much easier to mix the harder you stir them. It's exactly like how ketchup gets runnier when you smack and shake the bottle. Other broths are dilatant. They do the opposite. They thicken up and act almost completely solid under sudden impact or fast stirring, just like wet beach sand or cornstarch mixed with water. And finally, you have Bingham plastics. These act like a total solid until you push them with enough initial pressure, and only then do they finally flow. Understanding this physics is absolutely critical, otherwise we'd literally break our massive bioreactors trying to mix a liquid that suddenly decides to act like a solid.
So to bring it all together, back to our absolute baseline definition, everything we've just covered today, from the intricate cellular structure of bacteria and yeast to their rapid cloning processes, right up to the bizarre fluid physics happening inside giant bioreactors, it is entirely about how we harness and care for these living workers.
I want to leave you with this thought to ponder today. The everyday magic happening right in your kitchen, like yeast rising in a simple ball of bread dough, is the exact same foundational biological science being scaled up in massive industrial reactors to change the world. Thank you so much for joining me for this explainer. I really hope this deep dive has completely demystified the microscopic workforce all around us.